Good morning. Today's topic is genetics, specifically solving Punnett square questions. Alright, so our first question is going to be a basic monohybrid cross. You're going to see these a lot. Now, this one gives you all the information you need. So the question says, in guinea pigs, black fur is dominant over white fur. A heterozygous black male is mated with a white female. Then it asks you to give the genotype of both parents and give the possible gametes from each parent and the genotype and phenotypic ratios for the F1 generation. So we know a lot going into this. We know that black fur is dominant over white fur, and with that we're going to make a key. So what we're going to do is we're going to use a capital B for black fur, and then we're going to use a lowercase b for white fur. Alright, so now we're going to solve for the gametes. Because we know that the black male is heterozygous, it's going to be big B and little b. And the mother is white, so it's going to be little b, because that's the recessive gene. Now we're going to put these together in a Punnett square. So what we're going to do is we're going to make a box and we're going to divide that into 4 quadrants. We're going to put the gametes of the father on top, which is big B and little b. And on the left side, we're going to put the mothers, which is little b. What we're going to do is we're going to start crossing these together, and this is going to be the predictability of what the offspring will be. Now, as you can see, the genotypic ratio of the F1 generation is going to be 50% big B, little b, and 50% little b. The phenotypic ratio is going to be a little different. This is going to be whether it's black or white, and you're going to see it's going to be 50% black and 50% white. Alright, so we're going to do a little bit of a different problem, and I set this up already. As you can see, there are 2 black guinea pigs that are mated, and they produce 7 black guinea pigs and 2 white guinea pigs, and it asks for the genotype of each parent. Now, we don't know what exactly the genotype is, so as you can see with the father, we know it's going to have a big B, and the same with the mother, because they are both black. But the fact that they produced 2 white guinea pigs, they're going to both need 2 recessive alleles for that. So both of them are going to be heterozygous big B, little b. Moving on, we're going to see that this is a dihybrid cross. Now this gives you 2 different things. It tells you that black fur is dominant over brown fur and that normal length fur is dominant over short fur. Now we're going to set up our key because you always want to make sure you know what you're going to be using. So we're going to use big B for black fur, and then we're going to use little b for the brown fur. The same is going to go for normal fur. You always want to use the capital letter for the dominant allele, and we're going to use capital N for normal length fur, and we're going to use a lowercase n for short fur. So here we see that a male rabbit, which is heterozygous for black color and has short fur, is crossed with a brown female rabbit, which is heterozygous for fur length. So we're going to do the male rabbit's genotype, and we know that's big B, little and little n, because it has short fur, which is both recessive. Now for the mother, it's going to be brown, which is both little b, and heterozygous, which is large N and little n. So now we're going to find the possible gametes. For the father, that's going to be big B and little n, and little b and little n. And we're going to put that in our dihybrid cross, and we're going to put that on top because it's the male. And for the female, we're going to put little b and big N, and little b and little n. And we're going to cross these together, just the same as we did for a monohybrid cross, but now this time we're going to have 2 separate alleles that we put together. And so you're going to get big B, little b, big N, little n, big B, little n, and on the right side, little b, big N, little n, and little b, little n. And so we have a 25% chance of having big B, little n, which is going to be black and normal. Then we're going to have big B, little b, little n, and that's going to be 25% chance of being black as well as short fur. Next up, we're going to have little b, and big N, little n. That's going to be 25% of having brown fur with normal length fur. And then we have little b, little n, and that's another 25% chance. But now this is going to be completely recessive, and it's going to be brown and short fur. Alright, so now we're going to try our hands at an incomplete dominance question. So in roses, red color is only present in the homozygous RR, whereas pink is in big R, little r, and then white is completely recessive, little r, little r. So we're going to set up our key. Red is going to be big R, they gave that to us. Big R, little r is going to be pink. And the recessive little r is going to give us white. Now, because it's incomplete dominance, we're going to get that pink whenever it's heterozygous. So you have to keep that in mind whenever you're doing your Punnett square. So a white rose is crossed with a red rose. So that's going to be big R for the red rose, crossed with little r for the white rose. So if we're giving the F1 genotype and phenotypic ratios, we're going to have to cross those together. So you know the F1 is going to be completely heterozygous because you have two homozygotes that are being crossed. Now this is going to give you 100% big R, little r, which is going to be 100% pink. For F2, we're going to cross that big R, little r with another big R, little r, and that's going to give us a little bit of a different ratio outcome. So what you see, you're going to have big R together, big R, little r together, another big R, little r together, and then little r. This is going to give you a 25-50-25. So you can see 25% is going to be homozygous dominant, which is red. 50% is going to be heterozygous, which is your pink. And then the last 25% is going to be homozygous recessive, which is your white. So next up is going to be some sex-linked questions. So the gene for eye color is sex-linked on the X chromosome. Red is dominant over white. So when we do our key, you're going to see that red is going to be big R, and white is going to be little r. So now keep in mind that females have X and X, whereas males have X and Y. That's going to come into play when you only have alleles being passed down on the X chromosome. So males are only going to get one set of alleles on this cross. So here we have the father, and that's going to be X with a little r and Y, which is crossed with the mother, which is going to be X with a large R with another X with a large R. Now we're going to cross these together. You have X with a little r and Y with X with a large R and X with a large R, and this is your mother and father. And so we're going to cross these together, and you're going to see that some of them are going to be female and some of them are going to be male. Now these percentages are going to be specific to male and female. As you can see here, it asks you for the F1 male genotype, F1 female genotype, and the same with the phenotypes. So for the F1 male genotype, you're going to look at the Y column because only Ys make the offspring male. So you're going to have 100% X with a large R and Y because that's right there. For the females, you're going to have 100% X with a large R and X with a little r. This is going to be heterozygous because they receive both alleles. For the phenotypes, you're going to have 100% red as they only receive the dominant allele. And then you have, for females, 100% red. So if that was a little confusing, we're going to try and explain it with another sex-linked question. So again, the gene for eye color is on the X chromosome, and again, red eye is dominant over white eye. So we're going to be using the same key as we did last time. So again, that's going to be large R, which is red, and little r for white. And this time it's going to be a white-eyed female, which is mated with a red-eyed male. So when we're making the P genotype, we have to keep this in mind. As we see for the female, it is X with a little r and X with a little r because she's white, so she has to receive both of the non-dominant alleles. And for the father, we have X large R and Y because he's red and he only receives one allele, so that has to be the dominant one. So now when we're crossing these together, we're going to set up our Punnett square the same as we did last time, the same as we've been doing all video. And that's going to be with the father on top and the mother on the left. So as we're crossing these together, keep track of who is male and who is female and which alleles they receive from where. Don't put any alleles on the Y. That's a common mistake that people make without realizing. So now we're going to look at our Ys, and we're going to see that there's 100% of an X little r Y for the male genotypic ratio. And when we look at the female, we're going to look and see that there is 100% of X large R, X little r. For the male phenotypic ratio, we're going to see that all of the males have white eyes. And for the female, we're going to see that all of them have red eyes. And so that's going to wrap it up for today's video. I hope you guys understood a little bit better how to solve Punnett square questions. If you have any questions, feel free to ask, and enjoy your day.